Hello students, in our today's lecture, we will learn about mutations and its effects on our genetic code and about the tRNA, that is a transfer RNA molecule, which is covered in the chapter Molecular Basis of Inheritance. So let's get started. Students, what is mutation? Mutation is a phenomenon in which sudden changes in the DNA sequence takes place. Have a look at the image here. This is your original DNA sequence and this is a sequence showing mutation. Look at this point. Here in your original DNA sequence, you have a base that is thymine base, and in the mutated one, the thymine base gets replaced by a cytosine base. This change in the sequence of your DNA is called as mutation. Now, it results in the change of genotype, that is, it will change the character. Along with recombination, mutation is a raw material for evolution as it also results in variation. Students, variations are the new characters that get introduced into an organism because of the change in the original gene sequence. Students, let's have a look at the type of mutations. During mutation, possibility of loss, that is deletion, or gain due to insertion or duplication of a segment of DNA results in alteration in the chromosome. Students, have a look at this image here. It illustrates the different types of mutations. The first one, here you have this chromosome which has different genes from A to H. Now part of this chromosome is left out. Gene D is left out, and this process is called as deletion, and the resultant chromosome does not have gene D. So here you have a loss of the gene because of mutation, and it is altering the chromosome. The next kind of or the type of mutation is where you have insertions. Now again you have the same chromosome A to H. You have these two genes B and C. In insertion, what happens is it attaches to its sister chromatid in such a way that there is duplication of those genes. You can see here BC and BC is repeated twice. So this results in duplication of the gene on the same chromosome. This is a process called as insertion. Now, due to this insertion, you will see that you have a duplication of these two genes. So, because of this, there is a gain or there is an addition to the segment of your resultant DNA or the entire chromosome. Now, the next type is when the chromosome breaks off and reattaches backwards. This is a phenomenon where inversion occurs. See again, in the same chromosome, you have gene B and you have gene D this time. They get attached by altering their position or exchanging their position. That is, the D replaces the B, gene B, and gene B replaces the position of gene D. Again, this phenomenon also alters your sequence or the structure of your chromosome. The next type is when part of one chromosome breaks off and is added to a different chromosome. Now, till now, all the mutations that you are seeing or looking at are happening on the same chromosome. Here, in this case, two different chromosomes are used. So, when a part of it breaks off and gets added to a different chromosome, that process is called as translocation. See, you have the initial chromosome which is having genes A to H, and you have another chromosome having genes W, X, Y, Z. Now this region of the second chromosome, that is W and X, it gets exchanged and it is sent to your chromosome here which is having genes A to H. So now here your chromosome changes from A to H, it becomes W, X, A, B, C, D, E, F, G, H genes, and the second chromosome is left with just gene Y and gene Z. This type of mutation is called as translocation. So these are most of the possible types of mutations that occur. Now students, let's have a look at the outcome of mutation, that is how it affects our genetic code. Mutation can also occur due to change in a single base pair of DNA. This is known as point mutation. You have example of sickle cell anemia. Deletion or insertion of base pairs of DNA causes frame shift mutation or deletion mutation. So either you have an insertion or deletion of one or two bases, changing the reading frame from the point of insertion or deletion. Now to explain these points, I have put up an illustration. Have a look here. This is your normal DNA sequence. This is the mRNA which gets transcribed on the DNA, and these are the possible amino acids which get assembled after reading the codons on your mRNA. So this is a normal condition. Now we will look at what happens when there is mutation. Look at this DNA sequence. At this particular point, you will see that original DNA sequence had the C base, and here you have the thymine or the T base. Now so there is a substitution happening, that is C is getting replaced by T. Now as a result of this, the resultant mRNA, due to complementary base pairing, it will have A in place of G. So the codons now get changed, and because of that, in your original case you will see there's a different amino acid, and in this case where the mutation has occurred, there is a different amino acid. So this is substitution where a different base comes in, and because of which the codon changes, and then the resultant is you have a wrong amino acid coming. So this is how the change in your codons or the code that is there affects the resultant amino acid, and this is how mutation affects our genetic code. Students, the next example is, have a look at the DNA sequence here and compare it with the original one. You will see here at one point a new cytosine base is inserted. So you will see that instead of now your CTT being read, it is read as CCT, and this T gets shifted into your next reading frame. So students, because of this new base incision, again there is a change in the resultant codon which affects the amino acids that are coming or getting assembled during the process of protein synthesis. Students, the next example, see at the structure of your DNA. Here you will see that there is a deletion. One of these bases is removed. This A is removed here. So again, this process is called as deletion mutation, where one of the bases is removed out. So again, because of this, there is the resultant codons are changing. So there is, in both these cases of incision and deletion, there is something called as frame shift mutation occurring. Frame shift mutation is, students, where because of either an incision of a base or deletion of a base, the reading frame shifts. You can see, have a look at these arrows. It shows the change in the directions of your reading frame. Now, since the reading frames is changing, it's getting shifted from 3 codons each. This is a shift here in this direction, that is towards right. Here you have the shift in this direction, that is towards left. So as a result of this, the resultant codons change, and due to that, the resultant amino acids that are coded by these codons also change. And so this is the expected original amino acids, and you will see in each of this cases of mutations, you have different amino acids or wrong amino acids being coming, which are not expected in our original protein synthesis. And you will also note that the reading frame, once it changes, it remains unaltered from that point. The reading frame remains not just unaltered from that point, but from that point onwards. No changes possible once the mutation has occurred. Students, now let's have a look at the transfer RNA molecule. It's called as an adapter molecule. Scientists considered that there has to be a mechanism in which tRNA will read the codon and also simultaneously bind with the amino acid. So this is the structure of your transfer RNA. It's a two-dimensional structure. So there are two functions that your transfer RNA is performing. It has an anticodon loop where it recognizes the codon. At the same time, it has an amino acid binding site where it carries the required amino acid. Now, as amino acids does not have any special capacity to read the codon, so the tRNA molecule is considered as the adapter molecule. So your tRNA molecule is actually helping the amino acids to read the codon so that the correct amino acid comes in its place at the time of protein chain synthesis. This role of tRNA was understood much later. Students, let's have a look at the structure of the transfer RNA molecule. The cloverleaf structure. This is a two-dimensional structure of your transfer RNA. It possesses an anticodon loop here that has bases complementary to the codon that is present on your mRNA. These bases are called as anticodons. Now it shows an amino acid acceptor end at the 3' end having unpaired CCA bases. This is the 3' end. At this place you have unpaired CCA region where you have the amino acid that gets bind. So it is also called as an amino acid binding site. Now for every amino acid there is a specific tRNA molecule. So every tRNA molecule is specific as it carries a specific type of amino acid at its 3' end. The initiator tRNA is specific for methionine. Initiator tRNA, students, is the first RNA molecule which comes at the time of protein synthesis. It has methionine at its 3' end. So when you have your protein chain synthesis, the first amino acid that comes is always methionine. There are no tRNAs for stop codons. So stop codons do not have any tRNAs because it doesn't code for any amino acid, and that's how a stop codon stops the process of amino acid chain synthesis. Stops, or that process is also called as termination. This is the three-dimensional view of your transfer RNA molecule. Again, students, in the tRNA molecule, the actual structure of your tRNA molecule looks like an inverted L when it's in its three-dimensional form. Here again, I've put for you the two-dimensional view as well as the three-dimensional view. You can see here this is the anticodon region in the two-dimensional view and this is the anticodon region in the three-dimensional view. If you see, it forms an inverted L-shaped structure. This is its free 3' CCA end and this is a place where the amino acid is going to bind. So you have your messenger RNA having the coded information, your transfer RNA which brings the required amino acid, and you have the third type of your RNA which is the ribosomal RNA which holds the mRNA in place so that the 3 codons are read correctly during protein chain synthesis. So all your 3 types of RNA molecules are involved in protein chain synthesis, which we will see in our next lecture. Thank you, students, for participating in this lecture. We will meet soon again with the next topic. Till then, take care, be safe and keep learning.